Hello, my name is M.Grande, and I thank you for coming to listen to my presentation on the journal article titled Human Muscle Stem Cell Responses to Mechanical Stress into Tunable 3D Alginate Matrices. Now, to start, muscle injuries are the most common category of injuries in athletes and can comprise approximately 55% of all injuries that people can and will experience, on top of producing chronic pain, dysfunction, and recurrence. Now, for this study, let's start with the fundamentals. And the most important thing that we need to understand for this study is that human muscle stem cells are jobless cells that can change into muscle cells. So these stem cells are often looked at by scientists as plausible methods to treat muscle injuries and also a disease. However, they have one major limitation, which is their poor survivability after their transplantation into the human body. So they need a specialized environment. And the solution to this problem is physical calcium alginate hydrogels, which provide a habitable environment to them. On the screen is a depiction of a hydrogel. This study tests for three factors of these hydrogels, those being one, internal and external gelation modeling, two, their diffusion capability of small particles, and three, their impact on muscle stem cells after transplantation. For my review of literature, my first study shows that muscle stem cells are similar to previously known muscle repairing cells. My second article shows that muscle stem cells have been shown to increase muscle mass and muscle growth in patients with muscular dystrophy. Both of these studies have the same contribution to the present study, which is that it establishes muscle stem cells as a potential therapy for muscle regeneration. My third and final article shows that muscle stem cells have a suboptimal survivability after their transplantation into humans, which establishes the gap in our knowledge. How do we make muscle stem cells suitable for transplantation? The goal of this study is to test internal and external gelation-made hydrogels in their efficacy of transport for human muscle stem cells. The purpose of this study is to find a more efficient means for muscle regeneration. A possible hypothesis for this study is internal and external hydrogels will diffuse small molecules and minimally alter muscle stem cells. Now, finally, we get into the methodology of this study. Step one is to create the hydrogels using internal and external gelation. On the screen, there is a picture of how exactly we make these internal and external, but the main takeaway is that it's just two different ways of making the same thing that hasn't been tested before. The scientists will then test the mechanical properties of each of these. Step two is to evaluate the diffusion properties by placing the hydrogels in a chamber filled with a bunch of glowing molecules such as the ones seen on screen. After 24 hours, the glow intensity will be quantified, and that is how we will measure the amount of molecules that have diffused into the hydrogels. Step three is to gather human muscle stem cells and place them inside of the hydrogels and allow them to incubate. After 24 hours, they will be extracted and stained with coloring chemicals, and that will stain the nuclei either blue for alive or red for dead. On the screen is a depiction of blue versus red muscle stem cells showing how some of them are alive, some of them are dead. A short disclaimer is that it is a very simplified methodology which does not include replicable results. Now we get into the data and results slide, and this slide is purely for the gelation and modeling. So from the study, we learned that greater elasticity has been observed for external gelation and greater alginate and calcium percentages. As shown on the graph, the blue is always higher than the orange. That is, that the external is always more elastic than the internal gelation modeling. And that as calcium percentages go up and alginate percentages go up, so do the sheets, what it looks like, which is representative of the elasticity of the hydrogels. Now, two of these models, one that's internal and one that's external, showed very similar mechanical properties to natural skeletal muscle. So these were chosen as the models to test the impact on stem cells, which is 2 slides after this one. Now we get into the data and results of the diffusion. And we learned that both internal and external gelation can diffuse small molecules effectively. And the high elasticity models can even stop the flow of large molecules in. On the screen is a picture of how we measure this diffusion again with the glowing molecules. If there's glowing molecules seen on both sides of the hydrogels, then there is diffusion. But if the glowing molecules aren't seen in the inside of the hydrogel, then we know there is no diffusion. Now we get into the muscle stem cells impact. So think back, we have two models, internal and external gelation made, and they have very similar properties to skeletal muscle. These two were chosen, and muscle stem cells were placed inside of these hydrogels. And the results found that it didn't matter whether it was internal or it was externally made, and it didn't matter the time. Always over 80% of the muscle stem cells survived. And after they left the hydrogels, they still kept their ability to change into muscle stem cells. Analysis and discussion. So the data indicates 4 things. 1, that external gelation hydrogels were more elastic than internal. 2, alginate and calcium percentages affected mechanical properties. 3, internal and external gelation made hydrogels could diffuse small molecules, for example, nutrients necessary for muscle stem cells. And 4, that muscle stem cells showed no change in their ability to change into muscle cells after they were encapsulated in the hydrogels. This means that hydrogels are shown to be a viable means of muscle stem cells transport. Muscle stem cells are ensured safe travel that will not alter their structure, and they will have sufficient nutrient diffusion into the hydrogels. And the more elastic versions can also keep out large, harmful molecules. So for my conclusion, this study tested calcium alginate hydrogels' ability to transport human muscle stem cells. The findings include that greater elasticity was shown for external gelation and greater percentages of alginate and calcium. Internal and external gelation hydrogels could diffuse small particles, and muscle stem cells kept their functionality after their time spent in the hydrogels. Some sources of error might include that there was only an in vitro testing group, that is in a lab, and there was no control group. The applications of this study is a viable means of transport for human muscle stem cells, which means a viable means of injury therapy, which will improve overall wellness in the general population and in the sports circle population. Future research for this topic includes testing the ability of muscle stem cells to secrete substances out of the hydrogels, and in vivo testing, which is in a living test subject. Here are my references. Here are my references for my figures. And I thank you for listening to my presentation.